Here we are in part 5 toward the end of our presentation in chemistry, biochemistry, page 48, and we're looking at amino acids here. And so we have individual amino acids which taken together in a linear structure forms the primary basis for proteins or polypeptides. Over here we have them arranged with one of the acid moiety hooked up to the amino moiety. Here's this carbon, this acid carbon hooked up with the nitrogen. This is called a peptide bond, and we put them together slowly across. Basically, the genetic code for DNA is copied as an RNA, and then it's translated into a primary sequence of amino acids. And here I've made a little notation that if it's a true protein, proteins are big. They don't move through porous material very easily, and it's usually, I used to say that the NFL ruling here is that it takes about 1,000 amino acids to make a protein. Anything less than that, we're going to be calling it a polypeptide. There are some active polypeptides like insulin, for instance, or glucagon. These are going to be used in the body as hormones in the endocrine system. So this is the primary structure. We have altogether 20 of these, and we'll take a closer look at them when we get to metabolism and nutrition. But basically here we have the structure where usually over here on this side or at the very top in the Fischer structures, we have the acid moiety, which is a carbon with a double bond O and another oxygen. Over on this side over here, we have the amino group, and it's based on nitrogen. And if we're going to be hooking these guys up, we're going to be having this carbon lose the OH here and actually bind directly to this nitrogen right here, and that would be the peptide bond. Here we have a very simple molecule. It's called glycine. It's the simplest of all of the amino acids, and it's often seen quite often in things like tendons and ligaments. Okay, it's a very repeatable molecule, or in this case, a portion of a longer polypeptide. And so the side chains here are many and varied, and there's some basic ones, there's some acidic ones, there are some ones that are based on sulfur bearing, so we have that right there, and we have some that are relatively neutral as well. So 20 altogether. There's whenever we make a polypeptide or a protein, we have 20 different varieties of little amino acids. 8 of these are considered essential. That is to say, we cannot interconvert them in our own cells from one to another. Our cells are pretty smart that way. They're good biochemists, but they don't make some of the original amino acids, and so we're going to be learning the 8 essential ones when we get to nutrition. Okay, and then we consider what is the purpose of some of these little units. Before we get to that, let me just present you with this polymerization here. Do you see how we're creating like a thread-like device? And that's a very common thing. We create threads of protein in our body. Our hair, in essence, is one of those types of threads. The tendons and ligaments are made up of threads, polymerization of thousands upon thousands of amino acids. And basically, they're in a position so that their tensile strength is in this direction, right? And if we put a lot of these together, thousands upon thousands of these together, we can make a rope-like device like our Achilles tendon. It's quite substantial in its ability to bear forces and weight in a particular direction. Okay, then we also have helical structures, and then we have folded flat sheet structures, and then ultimately we get structures that can contain many of these ingredients, and we have what I refer to as globular structures. A large globular molecule then can have active sites within it, and so you can fit smaller molecules in this into the active sites, and this large globular protein can function like an enzyme, where it will collect small molecules, position them just the way you like it so that they can go together. For instance, you can put your glucoses together and make glycogen. Kind of a cool deal right there, and store it that way. Or you could take glycogen and then you can cut the glycogen into individual glucose molecules. So we have enzymes that are genesis enzymes that make things, and we have synthesis, or we have catabolic enzymes that break things down. Okay, so builders and cutters are what we have. And we also have some other things going on here. They actually make a notation here that some of our proteins are arranged in fibrous ways. Some of them are actually in sort of warped and weaved together into a fabric. So we'll be learning about that. There's what's called dense irregular connective tissue that underlines the outer skin of our body and the dermis. And it's a three-dimensional textile that has been woven with these fibrous collagen bundles woven together to make a nice, supportive, substantial fabric that underlies the skin. We then have the other consideration, which is globular proteins that act much more in the manner of enzymes, and they can hold small molecules together, or they can read something like a polymerase can read a DNA and copy it as RNA. So there's lots of features and sort of wonders of the protein universe. And it's protein really that makes us who we are. We have a genetic code, but we're made up out of protein based on the genetic code. And the protein also has, how shall I say, energetic and machine-like abilities. So we have some of our structure involved in the genetic code, right? So we can have a gene which codes for a protein sequence, an amino acid sequence. In some cases, the amino acid sequence folds and bends together in order to form a globular protein. So we might have some positive and negative charges on our amino acids, and they attract one another, right? And so they fold together to form this globular protein. And it does work for us. So that concludes this particular chapter. Let's actually make just a few more notations here. Since we can't heat anything in our body, we can't cook anything to make anything go forward, we have what's called the heat of the reaction here. Instead, we're going to have to use reduced heat, and we're going to have to use a helper, which is an enzyme. And so we can actually create reactions. We can put things together, or we can break things down to lower oxygen or oxidation levels, it's referred to. So high energy bonds to low energy bonds. There's that type of thing. Here we have a globular protein with active sites in which two smaller molecules get together in the active sites, and they're positioned just right so that you can actually affect a bond between the two, and you end up with a product at the end there in which you have a new molecule. So that's the idea. This is the final category right here, the DNA. And this is a sequence of 4 different base pairs. And we actually have, we take them 3 at a time. They're called codons. And they code for individual amino acids. And so you can read through a gene, a genetic region here of maybe 1,000 or even 3,000 different base pairs. And as you're reading along, if you take these base pairs 3 at a time, you're actually reading the words, as it were, or the amino acids of a protein or a polypeptide. So you can read that. I've seen there's machines that do that, and they advertise in Cell magazine, and they have little advertisements sometimes. And one of them, I remember, advertised their gene reading machine and said, "Read any good DNA lately?" So that was a little reach out there to the lab professional who would like to sequence DNA and see what's going on. Okay, so we'll bring that up again from time to time. Okay, that concludes the this particular playlist on chemistry, biochemistry.